Okay. So, yeah, please let me know if you guys can you, has anybody tried to use that YouTube? No? Okay. Would somebody be willing to stick a few minutes back and we can see what it looks like from your side? Okay, thank you very much. Yeah, I got to remove all those videos. Yeah. Yeah. Apparently, I had a whole thing going on where the classrooms were taking parts of my other storage, and instead of just incorporating the links, even from my external accounts, they were actually taking the stuff and making copies in every single class. And so, like, part of my research folder got copied, and then I had a bunch of video copies. And then the classes that I delete on the Google Classroom don't delete in the drive. Yeah. And yeah, so I was like 500 gigabytes apparently. I never even realized. Yeah. My research folder alone is like 100 gigabytes. If you copied that fucking thing, that'd be crazy. Okay, so this is where we had left off last time, and you can see the substantial conformational changes that we associate with the myosin transition state here of binding. Remember, we're binding in this myosin case with the ATP. This is going to drive that ATP hydrolysis, a conformational change where we are literally moving something along in physical space, right? This is a mechanical engine, okay? And you can actually see it looks like a pedal crank. Everybody see that? You know what a pedal crank is? Something with like a pedal on it that can move stuff around, or you can attach things to move. Okay. So we can actually see here that these relatively minor small changes that are in the active site actually causes fairly large changes over here, right? Okay. And that's actually what an engine ultimately does, doesn't it? It transfers a relatively small change at the engine site to a large change somewhere else, doesn't it? Okay. Okay, you can actually tell this fairly easy if you look here. See the blue corresponds to this state right here. You can see that that's actually been sucked into this active site more. So you're having more binding, right? And the red here is the other one that's a little bit further away, and then it has this part that's swung up there. Okay, so the altered conformation of your myosin is going to persist for a substantial period of time. The myosin is actually a fairly slow enzyme. This has a typical turnover rate of about 1, so this would be 1 hertz, or 1 over a second, okay? Happens once a second. This is the same thing as you would see like an RPM. Do you know what an RPM is? Rotations per minute, and it's for your car, right? And literally how many times it would rotate per minute, right? Okay. So the isotopic labeling studies that use the water that's labeled with the O18 were conducted in this case. And this is why you're seeing these things, is this is a good book that's meant to take you all the way through your professional careers, okay? And that means that, yes, while it has explanations for each thing, it also has actual research backing every single thing that it states, right? And this is actual research, right? And that's what that looks like. So we have the isotopically labeled O18 in this O18 water, isotopically enriched water. This is going to attach to the phosphate here. And what we're seeing is we're experiencing a hydrolysis of this phosphate, which leaves you with the ADP. Okay, and everybody's familiar with the ATP, ADP, AMP terminology? Fantastic. Okay, so it's expected the phosphate would contain 1 oxygen derived from water and 3 initially present in the gamma phosphate of ATP. The study actually reveals, gives results that reveal that 2 to 3 of the oxygen atoms in the phosphate were actually derived from the water. So this actually indicates that the ATP hydrolysis is actually readily reversible. So when we actually looked at the results of this study, this is the expectation, okay? That you have a hydrolysis, and that would give you one isotopic 18 labeled phosphate, right? And you'd have one oxygen that had been added to it, right? But that's not what we got. We got 2 to 3 of those oxygen atoms getting replaced on that gamma phosphate, okay? What does that tell you about this? Well, it doesn't just do it once, does it? We're actually getting these happening over and over, and so we're actually getting a lot of replacements occurring, okay? And again, this indicates that ATP hydrolysis is rarely reversible, that the rate-limiting step in the hydrolysis appears to be the phosphate release of the hydrolysis. This is actually the reversible hydrolysis here that you're looking at. And so what we can see here is that we have the phosphate binded, water comes along and does the hydrolysis, right? And then we get a rotation of a new phosphate in, whatever phosphate happens to be around, okay? And then it's going to have a binding going the reverse direction. Now, because we had a rotation of the phosphates in, there's a very high chance that one of the other oxygens is actually going to be in the spot that would be the hydrolysis spot, right? And so what we'll see is that over time, you're going to get more and more of that enriched isotopically labeled oxygen attached to that last phosphate, aren't you? Okay? Anybody have any questions about this? No? Fantastic. Okay, so the actin filament is going to have a polymeric structure here. And so the actin filament is going to form, actin will form the filament along with myosin, and this can then move because that myosin is the engine driving our actin, which is kind of our axle or shaft, right? Our structural component of this. Each actin monomer is going to contain a bound nucleotide, ATP or ADP. And so right here we actually see the pointed end here. This here is the barbed end. This one is the nucleotide binding site right here. Okay? And notice again, this is that twisted dimer structure. Everybody see that okay? That helical structure? Fantastic. Okay. So we can watch single molecules of myosin moving along the actin filaments. The myosin can be labeled with fluorescent tags so they can be localized. In the absence of ATP, the myosin is going to remain fixed in place on the actin filaments and isn't going to move along it. And so this is an abstraction of what they learned from those labeling studies. Their labeling studies don't have nice figures like this. It looks more like a rod, vaguely grayish rod shape, and then you have a slightly differently colored, vaguely grayish blob, and that moves along it, right? Okay. I know, it's not exactly so pretty, is it? That's much prettier. Now, in the presence of your ATP, the myosin molecules are going to move along your surface, and this movement is going to be powered by the ATP hydrolysis. This is actually your position in nanometers, so this is starting from your 0 position where it started, right? And then as we go along time, what we see is we actually have a stepped structure. Can everybody see that okay? How it's stepped? Okay. And then what we see is that it's going to transfer position over time. What do you think we're seeing here? We'll pause here a little bit so you can have an opportunity to expand your ability to think in a way that is scientific, right? To use your critical thinking skills. What do you think we're seeing here? If you had to guess, which I'm making you guess. All right. So when we do a step right here, what does that tell us when we go vertically? That's when we're changing position, isn't it? When we're flat, we're not actually moving, are we? Okay, we're just waiting. So we wait, move, wait, move, wait, move. And we keep doing that over and over, right? By the way, notice that is exactly like your engine works. Your engine also does the same thing. So does electricity, by the way. Yeah, exactly. We have to wait for the ATP to come in, right? We do the ATP, we do the reaction, we get the movement, okay? And those are quick. They take very little time, right? If you were to zoom in far enough, and you can kind of see it, it has a little bit of over, but not a lot, right? These are happening real fast. And then we have to wait again for the exchange, and then we do it again, right? And so that's actually what we're seeing here. This is the myosin dimer right here. This is your catalytic domain down here. This is your actin filament that we're going to move along. Remember, that's like muscles work, right? Okay, because it's muscle. That's how it works. Okay, it's literally like a bike. So you pedal, you grab your chain, right? And you move the, oh my God, I'm having a blank spot right here. The gear, excuse me. One notch, right? Along the chain. Okay? And then you do it again and again. And in fact, if you paid very close attention to a bike, you would also get a similar stepping, which is you literally taking steps downwards, right? You moving your pedals with your feet. Push, 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 push. Yeah. So now what you're seeing here is if you looked at these ups, these are about 74 nanometers each, and that's the distance that these catalytic domains go each time. Okay? That's that swing each time. That making some sense? Fantastic. Now notice, by the way, this is a dimer here. Again, you see the dimer structure? That we have two of these. And so it's literally, it's very similar to like if you had a bike that only had two teeth on it, right? Move, and then the next one attaches, is the one on the end, move. And then each time it just moves the chain, right? Okay, so this is an abstraction of this that will help you understand this process a bit more. Remember that this would be one of the monomers here, right? Okay, so here's the overall mechanism of that myosin movement along the actin film. Remember, this is going to be our monomer here, our yellow one, and this is going to be the process of us moving along this. Okay, so right here we have the myosin. It has not had any ATP bound to it yet, right? So we have actin, myosin, they're attached, no ATP yet. So what's going to happen is our ATP is then going to bind. This is actually going to detach the myosin from the actin, right? Then we're going to have hydrolysis. This will leave us with ADP and the phosphate still attached to the myosin, okay? This is then going to reattach at a further distance. Remember, we detached. We have the option of moving closer or further. In this case, we'll move further because it will be energetically favorable, okay? And then once we've moved further, we're then going to have this reattachment while the ADP and phosphate are still attached to the myosin. But we have done the hydrolysis at this point, okay? So this has changed the structure due to that change in hydrolysis. Now, what's going to happen next is we're going to again reattach here. We are then going to lose the phosphate. Now remember, our phosphates can come back on, right? So keep that in mind that this phosphate can come back on with a slight rotation. And we've actually seen that in the previous example. Remember where we got the 2 to 3 oxygens that were labeled? Yeah. That's what we were seeing. It can come back on here. Okay? Okay, so what happens next is the ADP is then going to be released. This will reset our myosin, and then we start the process again, right? By the way, notice there is not an arrow coming back onto here. Why is there no arrow coming back on, making it look like a cycle? I mean, it's cyclic in process, right? Are we actually resetting to the exact same states? No, we've accomplished work, haven't we? Yeah. We've moved, well, approximately 74 nanometers, right? Okay. And so we don't actually reset, do we? Yeah, we lose the ADP and we can take on new ATP, but we're down the chain. Okay, anybody have any questions? Fantastic. All right, so the myosin assembly is from the muscle, has a two-headed structure. The active myosin motor system in the skeletal muscle will contain these elements, which are the organized assemblies of actin molecules. These are going to be our thin filaments, are going to be the organized assemblies of our actin molecules. Those are these spider web sort of looking things over here, where it comes frays apart. Okay. We have the multi-headed assemblies of the myosin molecules. These are going to be our thick filaments that form our thin filaments. Then we have the myosin that moves along the actin filaments to cause muscle contraction. Okay, so keep in mind, so when we're looking at muscle down on this thing, okay, the thick and thin filaments are going to be from our visual perspective, okay? So the parts that look thin to us is going to be this part right here, where they're braided together and stretched out, okay? The parts that look thick to us, that are very colorful, are going to be these parts right here, which are our rays, the multi-head assemblies of our myosin, right? I think that does a better job of clarifying that, right? Okay. Did I lose anybody in that? Fantastic. Okay, so this is actually going to be the actin networks. We've imaged these using modern fluorescence microscopy. When you see these diagrams, by the way, don't be fooled by them. This is not actually what you would see if you looked into a microscope, okay? This is what you see after a computer gets at it to make it something you can see, okay? But they're very, very highly related to actually what you would see. It's not like it's complete fabrication, just it's colored better so you can actually see stuff. Okay. So here we have the cytoskeleton. This is going to be your internal cellular network. Cellular skeleton, it's a better word probably, containing the actin filaments. And you can see these actin filaments, can't you? Can you see that okay? You can see how they braid up into connections, and then eventually they braid up into further connection. And that gives you your overall tendons and muscular structure. And you can start kind of seeing the overall muscular structure, can't you? Yeah, which is a very nice diagram. So this is going to play a role in determining cell shape as well, because remember, the actin filaments are actually the structural components of a cell, aren't they? And so these are also the structural components of your cell. This is what you would move stuff along in your cell. Remember your cell cycle, where you have the actin filaments, and then you're going to move the parts of the cells. This is also what you're using there, okay? Same sort of assemblies used in all these different purposes, right? Okay, and that is the chapter. Let's go ahead and get on to the next one, I guess.